African trypanosomiasis. And there are two versions of this disease. So in West Africa, you get a form caused by Trypanosoma brucei gambiense, but the form that we have here in East and in Southern Africa is called Rhodesian sleeping sickness, or Rhodesian human African trypanosomiasis, and is caused by Trypanosoma brucei rhodesiense. And this is carried by wildlife in particular, and sometimes also livestock. So it's found in particular in and around wildlife protected areas where the wildlife and the tsetse are found. And then there's the animal form of the disease. So the same species, Trypanosoma brucei, as well as other species, can cause disease, particularly in cattle, but in other species of livestock as well, and has very significant impacts. So estimated to be around $1 billion a year lost in direct impacts, and up to $4.5 billion per year lost in the indirect impacts from not being able to produce livestock in some of these areas where tsetse are found. So really significant disease in terms of impacts on things like livelihoods, poverty, food security. Diseases, and particularly diseases in animals, have really been part of the history of Serengeti. So just looking back into some of the history around East Africa in general, but including in this area. So if we go back to the 1880s, rinderpest, which is a virus that particularly affects cattle, was introduced into the northeast of Africa, probably into Eritrea, and then spread very rapidly over 2 decades down through the whole of Africa, right down to South Africa. And it killed 90 to 95% of the cattle. So that's an enormous impact on people and households and the environment. And it also killed many wildlife species. And that led to very significant famine and starvation. In some areas, it was estimated that maybe 2/3 of the population died from starvation. And that had really significant impacts on tsetse and tsetse distributions as well. So initially, the numbers of tsetse dropped because there were no hosts to feed on. But then rapidly, the bush extended into all of these areas where there were no longer cattle, other livestock and wildlife. And then that tsetse population then spread very fast. And so many areas which had previously been clear of tsetse became really areas of very heavily tsetse infested bush where many remaining people and animals were getting sick. And that led to very significant outbreaks of the human disease of sleeping sickness from around 1920. And that again caused very high numbers of deaths in some of those areas. And it was only really later in the middle of the century when a lot of control was done in terms of bush clearing and in many cases moving people out of those affected areas that they managed to bring those epidemics under control. So we cannot forget that the human disease can cause these very significant epidemics and impacts. But we also see these impacts in the environment and the ecology here in Serengeti. So the buffalo and the wildebeest populations, for example, took decades to recover from the population crashes caused by rinderpest. But probably also the invasion of tsetse and the expansion of bush at that time also protected the ecosystem from settlement until it was protected and gazetted as a national park. And I think many of you here will know better than me the ecology of the relationships between cattle grazing and fire and tsetse and bush encroachment. But tsetse are a really key part of that, both in terms of historically being a reason that people wouldn't go into or take their animals into tsetse areas, although that really has changed now that people have access to control methods to protect their cattle, but also the patterns of tsetse invasion when you get expansion of bush. And again, I think a lot of you probably are familiar with tsetse when you're moving around and being bitten by tsetse, but perhaps not so much with the ecology. And tsetse are really a really interesting insect. So they're quite unusual in that their strategy for reproduction is they just produce one live larva at a time. So that's very different to many fly species. It's something in ecology which is called a K-strategist. And that gives them some quite particular features which are important when we think about control, because the population only grows slowly because each female is just producing one larva at a time. They can also move really big distances. So I've put that they can move maybe 1 kilometre per day. But actually, if you stretched out, they do lots of little moves. And if you stretched out all of those moves, they can actually move about 10 kilometres in one day. And they can fly really fast, as many of you will know, because you see them flying alongside your car. So maybe 20 kilometres per hour. So that means that even if you're clearing tsetse from an area, it's quite quick for them to re-invade again because they can fly fast for quite long periods a day to move into that new area. And if you clear an area, the speed of the tsetse front moving back into the area can be 25 kilometres per year. So they can reclaim a cleared area of 25 kilometres per year. So we can think about these when we're thinking about tsetse control. So there have been some very large-scale tsetse control programs, and they can be very effective. So, for example, in Okavango Delta in Botswana, they carried out sequential aerial spraying. So that's spraying of insecticides from a GPS-guided plane. And because of this strategy of reproduction where they reproduce slowly, it's possible to do that at sequential intervals, which are sufficient to control the tsetse, to cause mortality in the tsetse population, whilst not affecting all of the other insects. Because obviously if you're spraying insecticides, you're worried also about the impact on biodiversity more generally. But because other insects usually don't use this strategy, you can time that spraying to affect primarily the tsetse. And Okavango Delta still has no tsetse in that area after that spraying program. Another example of large-scale control is when you irradiate male tsetse flies so that they are sterile and then release millions of them into the environment so they mate with the female flies so they cannot produce offspring. You have to release millions, and so far the only successful example of this is on Zanzibar. But it's something which can be very effective if you've got a small population of a single species. But obviously it's very prone still to that problem of reinvasion. So it worked very well on Zanzibar because it was a small population that had already been reduced through other control measures, and also because it's an island, so there is nowhere for reinvasion. It's quite hard to achieve this, and there's not really any good examples yet of where this has been achieved in an area which doesn't have natural boundaries like that, and particularly if there are multiple species of tsetse, as we have here, for example, where there are 4. So targets, so insecticide-treated material in the right blue and black colours to attract tsetse, and there's been decades of research to design the most effective colour and shape of targets to use from that. I know you are very familiar with because there are many around here and many of you spend a lot of time putting these up, and they can be really effective as long as they are designed and made correctly to those specifications, and as long as they are resprayed frequently with insecticides and then maintained. The problem is that often that is not done. Often when you go to other areas, for example, you might see in the national park, you often see some old targets that are tied up in a tree, that they will not be doing anything. They have to be standing clear and made exactly to the specifications and maintained carefully. But if you manage that, then they can be very effective at tsetse control. The other approach is if you're in an area where there are cattle, if you insecticide treat your cattle, then your cattle are essentially acting as moving baits, a little bit like a moving target. They're drawing the tsetse will be attracted to the odour of the cattle, and then when they come in to bite them, they are killed by the insecticide. So obviously this doesn't work if you're in a protected area where grazing is not permitted, but it can be very effective in areas that cattle are present. But always there is this problem that reinvasion is always a challenge. So it's often having to think about buffer areas or continuing control to prevent reinvasion. So just coming back a little bit to the human disease in Serengeti, and I just wanted to highlight this in particular because I have heard sometimes people say that there is no human sleeping sickness in Serengeti. It's not a problem here. It's not common now, but it is there. And it's important if you're working in this ecosystem to be aware of that. So I mentioned previously that there had been these very big outbreaks from the 1920s with very high levels of mortality. They were brought under control by about the 1950s through particularly bush clearing. And then through that period, from about 1960s up to 2000, there were a small number of cases ongoing, but not very many. And really people have become quite complacent until 2000, when there were about 25 cases or so in 2001. in the Serengeti ecosystem, particularly being identified in tourists, which highlighted that there was increased transmission going on. We still don't really understand the reasons for that epidemic. And since then, there have been ongoing cases. So just to show, this is cases just in Serengeti since 2000. So you can see this little epidemic in 2000, 2001, and then you can see the ongoing cases. And just to highlight here, the orange colours are cases that were in tourists and were diagnosed overseas. And the blue cases are ones that were diagnosed in Tanzania. These are all published cases. And you can see in particular that there are very few cases in Tanzanians that have been diagnosed in the last few years. One of the problems is, it used to be when you would go to the clinic because you had maybe fever, they would take a blood sample to do a blood smear for malaria, and at the same time they would look at that blood smear for trypanosomes. But now when you go to the clinic, they do a rapid test for malaria, and nobody does a blood smear for trypanosomes anymore. Not just here, but across many areas of sub-Saharan Africa. So the number of cases of sleeping sickness that are being diagnosed has really dropped. But part of the reason for that is we have stopped testing for it. So now we have very little information about the true burden of disease which is happening. And it's important for you to be aware of that risk if you are working in areas where the disease is found. So I'm just going to talk a little bit more about the research that I've been involved in over the last 20 years, which really started looking at trypanosome transmission in wildlife. So thinking about how the human infective trypanosomes exist in a wildlife ecosystem, and then thinking about what happens at the interface when we move out into the farming areas, and particularly about the role of cattle in terms of reservoirs of human disease, and then thinking about actually the impacts of disease in livestock and what farmers are doing in terms of disease control in livestock. So thinking first just about the situation in wildlife, one of the first questions that I looked at was if we're thinking about the human infected disease, so the trypanosomes that infect people, which wildlife species can actually carry that species? So I spent a lot of time collecting samples from different wildlife species to look at prevalence. I spent a lot of time working on warthogs, as Matt remembers, because we were catching warthogs up here. And actually we found the prevalence in warthog and buffalo to be relatively low. But we found a really quite high prevalence in, you can see to the right, things like impala, reedbuck and bushbuck. But it's not only important to think about which species are carrying these trypanosomes, but also which species are being fed on by tsetse flies. So we also looked at lots of tsetse to see, we can take the blood meal from the tsetse and we can see what they have been feeding on, which animal species are there. So you have two main species of tsetse here, Glossina swinnertoni and Glossina pallidipes. So I've put in some pictures here, they're not great, but the swinnertoni is the smaller one, and the bands on the body are a little bit more distinct. It is more like black and white. And the pallidipes are quite a bit bigger, and it's a little bit browner. The stripes are less distinct. And usually if you're driving around in the car, it is the Glossina swinnertoni that will be following you around because they are attracted to moving objects. The pallidipes usually hang out in trees, like in shade, in trees or bush, and wait for hosts to come to them, and then they will go in and feed. So when we looked at feeding patterns, you can see, so the blue swinnertoni, the biggest amount of feeds is coming from warthog, which is why we were particularly interested in warthogs, and then from buffalo and giraffe. But they will also feed on really whatever else they can find. So we also found things like elephant, hippo, eland, for example. Pallidipes are a little bit different. So they are feeding predominantly on buffalo and then followed by giraffe, elephant, warthog. So we have slightly different feeding patterns between those two fly species. And then we can combine all of this information together into a mathematical model that looks at disease transmission to say, if we combine this data on prevalence with this data on feeding patterns, which species actually contribute most to the transmission of Trypanosoma brucei, which is this trypanosome species, which includes the human infective subspecies. And interestingly, we actually, it comes out that buffalo are probably really quite important. So you wouldn't think that when you look at the prevalence data, but because they are providing very many of the feeds And because also there are quite a lot of buffalo, we're actually seeing that they are quite important. But warthog and bushbuck obviously really important as well. So it's a combination, it's being driven by a combination of those feeding patterns, which hosts are being fed on most frequently, but also which species are infected. And I should say that we also measured the prevalence of human-infected trypanosomes in the tsetse, and it is very low. So when you're being bitten by a tsetse and you're thinking, oh, I wonder if this tsetse carries disease, the frequency is very low. It's maybe 1 in 10,000 flies. So you have to be very unlucky to be bitten by a fly which carries a human-infected trypanosome. But obviously some people are still bitten by one. So we then looked at the question of, if we know this is what happens in the wildlife, what happens when we move to the edges of the protected area? So we were concerned at this stage that possibly cattle might be acting as a reservoir host for these human-infected trypanosomes as well. So we did a lot of sampling. So you can see the cows show where we were sampling cattle in herds around the borders of the protected area. The blue are where we put tsetse traps up like this. You can see that we had 2 transects in Ikarongo, one in Grumeti and one going through the open area through Rabanda and into the park. So they were all measuring how the numbers of tsetse varied as he moved on a transect from a wildlife-dominated system out into a livestock-dominated system. And so what we found from this is that the number of tsetse drops really fast when you move outside the protected area. So this shows 5 kilometres inside the protected area. This is the boundary, and then 5 and 10 kilometres outside. So as you move out into the farming areas, the number of flies drops very quickly. Now that doesn't seem that surprising. I think that we see that anyway. As you move from the protected area to the farming area, there are less flies. But we were interested to understand why that is the case. So we put together a spatial model to predict where we would expect to find tsetse. So if we think when we're moving out to the farming areas, the habitat has changed. So we assumed it would be driven predominantly by habitat. But actually, in these areas outside the game reserve boundaries, there are many areas of nice tsetse habitat where we would expect to find tsetse, and we still don't find them there. So we were curious as to what is the reason that you don't find tsetse outside of the protected areas. And then we started collecting data on insecticide use by livestock keepers. So livestock keepers use insecticide on their cattle really frequently. So we found, for example, in surveys in Serengeti district, but we've also done it in other areas, that perhaps 1/3 of livestock keepers will report treating their herd within the last 2 weeks. And we did some chemical analysis of this so that we could test that and see whether that's what they were telling us really matched up with reality, and found relatively similar results. So these will test the presence of cypermethrin for about 2 weeks. So again, we found about 1/3, just under 1/3 of herds that had detectable insecticide. So we can see that there's really quite frequent insecticide use, and that's through dipping and spraying of their cattle to protect them against tsetse, but also to protect against ticks. And again, we could combine all this data into a model to try and explore what that insecticide means for transmission. And what we could find is that that level of insecticide use is causing essentially control of the tsetse population. So the reason that we're not finding many tsetse outside the park is partially because of the change in habitat as you move to the farming areas, but it's partly because there's so much insecticide being used on cattle that that's actually controlling that tsetse population, increasing the mortality of the tsetse population. So essentially what livestock keepers have done, just through their own reasons for treating their animals, is implement the method I mentioned earlier of insecticide-treated cattle, because these cattle are treated with insecticide and then are moving around like moving baits. And what we can see in the figures here is if we look at the particular trypanosome species, so if we look at what that means for transmission of the trypanosomes, that we see an enormous drop-off in the transmission of our trypanosome species once we move outside of the park boundary. And we can see that in particular for the species which includes the human-infected trypanosomes, because it's just less transmissible, so it starts at a lower level. So what we can see is that actually farmers are doing control to protect their livestock from the cattle trypanosomes, they're not aware mostly about the human disease risk, but they're actually achieving a reduction in transmission, particularly of the human infective Trypanosomes too. So this is a really nice example of control aimed at a livestock pathogen, but it's also reducing the human disease risk. So we think there isn't an increased risk at that boundary because of this level of control. And then just to mention briefly some of the work that we've been doing with livestock keepers. So that work on insecticide use made us think more about what control measures livestock keepers are putting in place to control animal trypanosomiasis. And these measures towards the right, so I mentioned the things like the sterile insect technique and aerial spraying, can be really effective measures of controlling animal trypanosomiasis, but they're really hard to put in place. They require big, centrally coordinated and funded projects, which are becoming rarer and rarer. So what this actually means is that the burden of control of animal trypanosomiasis really sits with livestock keepers, and the tools that are available to them are the use of trypanosides and the use of insecticides predominantly. We know that livestock keepers use trypanosides very frequently. So we've done work here in Serengeti, but also this was done in Pangani district, where there are mixed livestock systems, including dairy, and in Simanjiro, along the edge of Tarangire, so pastoralist systems. And we find that the rate of use of trypanosides, for example, in Pangani, people were treating each animal almost every month, so 10 times per year on average, and 4 times per year in Simanjiro. So really a lot of time and money being spent giving trypanosides, and a lot of these drugs being used in the market. But what we were finding is that when we held workshops to talk to farmers about some of the challenges of animal trypanosomiasis control, they would very frequently report that the trypanoside drugs, so this is things like diminazine and isomethamidium, were not effective, and they were increasingly seeing a problem with these drugs not working. So we then were able to get some funding for a big project that looked essentially at quantifying, is that the case? Is it true that some of these drugs are not working? And what are the reasons why drugs are not working? What are the reasons that we're getting failures in treatment? So we conducted a study in Serengeti district with livestock keepers, where we recruited 21 farms, and we had 30 animals on each farm. And essentially each time a farmer went to use one of these drugs, we would document everything about it. So we'd document how he gave the drug. We would take a blood sample beforehand so we could look at whether the animal really was sick with trypanosomiasis. And then we conducted follow-up sampling to look at whether the animal then recovered. And my colleague Paul here was the one who was leading this fieldwork in Serengeti, which was a very intensive study and being ready every time a farmer called to run and collect this data. So it's a very nice data set, and nobody else has ever really used this approach, but it worked very well. So I'll just talk about a couple of the findings from this study. So we found that around a third of treatments were not given properly. So livestock keepers almost always are injecting these drugs into their livestock themselves. And we found that we looked at a number of different reasons why the drug might not be given correctly. So we looked at dose, whether they're injecting in the right place, whether they're giving the injection competently, and whether they are storing the drugs correctly. And the most common reason for the administration to be inadequate was that the dose was incorrect. We analysed all the samples to look at when we get failures in treatment. Some of these drugs are used to prevent infection, and we found that in that case, actually, we really found quite a lot of failures. So around 20% of the use of these drugs was not successful. And when we looked at the causes for that, maybe a third of those could be associated with times when the drugs were not given correctly. But that leaves 2/3 of cases where there was no obvious reason why the drugs had not worked. So we looked then at whether the drugs were of sufficient quality and also whether there's resistance present. And we found both of those things to be the case. So just finally, to give you an example, we've done quite a lot of work resulting for that, looking at the trypanoside drugs, which are available for sale in the livestock agrovet shops, in the livestock markets. And we commonly find that there are a lot of those drugs where when you test the sachet, there is no drug in there. So there are lots and lots of counterfeit drugs that are circulating. And we found, you can see some of the results here on the right, in some districts that we found almost half of the isometamidine drugs for sale contains no active ingredient. And there's a couple of examples here of counterfeit drugs that we found. So this is quite a serious problem, and livestock keepers are very concerned about this because they are spending money on these drugs, and actually sometimes there is just no active ingredient there. So I'm going to stop there so that I can hand over to these guys to say a little bit more about the next steps. So just to say, I've talked about some of the work that we've done, but there are so many more questions that need answering on different aspects of the system. So just thinking about the transmission within the wildlife systems, I talked about work looking at Brucei, but actually we understand very little about the particular subspecies which causes human disease and how that in particular emerges and spreads. We're increasingly hearing examples of introductions of wildlife between, or movements of wildlife between protected areas, not just in Tanzania, and we've been working quite a bit in Malawi as well, where this is happening pretty commonly. And there's some big questions around how that impacts on trypanosome infections in wildlife. We looked at the role of cattle as potential reservoirs of disease, but we haven't really looked very much at disease in sheep and goats. And there's a number of questions about how we can best support livestock keepers in terms of the control measures that are available to them, and particularly understanding more about these issues of resistance and treatment failures. So we have lots more questions. And just my final slide, just to leave with a couple of points to think about. So I think tsetse are often described as the greatest conservationist or the greatest ranger, because they have that role for a long time of protecting protected areas, and meaning that people didn't want to come in or bring their livestock into protected areas. I think that's not the case anymore now that livestock keepers have more control options available to them. And now predominantly it's just a major cause of impacts on livelihoods and poverty and human health. As I mentioned before, really just to highlight to people working around wildlife areas that sleeping sickness is a disease which is present. It's not common, so I'm not trying to scare people. It's only there in a very small number of tsetse flies, but it is important just to be aware that it does exist. And if people have signs which are similar to malaria but are not resolving, you always need to ask to be tested for trypanosomiasis as well. And finally, just to comment on the sort of work we've been doing with the livestock, we've got sort of more research questions that we want to answer on this, but we're also thinking about how we best talk to livestock keepers about some of these findings. So we've done some small scale training and information with farmers, mostly our study participants who are really engaged and interested in more information about this. So we're thinking about how we can build on that in terms of the information that we give to farmers and the routes to do that, but also possibly how we engage more with women in livestock owning households who often miss out on the sorts of training opportunities that might be provided. So I'll stop there. Obviously, I've covered, it was a bit of a whistle-stop tour of lots of different bits of research there. I'm very happy to talk to anybody afterwards about any of the specific things that I was covering. And obviously, I'm talking on behalf of a big team of people who've been involved in all of this. I'm going to hand over to Paul. He's going to talk about the next bit.
So thank you very much, Jared, for taking us through the main topic of today. And so I'm going to present to you a little bit about what I'm going to do in my master's degree. And as Harriet has said, we have been focusing on cattle and living sheep and goats. So I'm going to investigate the role of small ruminants in transmission dynamics of African trypanosomiasis, which includes both the human infective, but also the livestock, which is called animal African trypanosomiasis. So my name is Paul Sampson, as I've been introduced. But before I proceed, I should just mention a little bit about my background. So I started in Sambara Primary School. That was 2006. I graduated. Then I went to Ihungo Secondary School, which is in Bukoba. I studied 10 subjects there, so including French. But when going to to Form 3, I was allowed to take only 9 subjects. So I was weighing because I was the top in French, and I was doing well in agriculture. So I started weighing. Then I loved to go to science. I left French. Okay, so then from there I graduated in 2010. Then I went to Kiba Secondary School. Then I graduated. I went to Sokoine University of Agriculture. I graduated in 2018. I took Bachelor of Veterinary Medicine. Then after that, I worked in tsetse and trypanosomiasis projects, including combat, Harriet has said about it, enables, and other projects. So then in 2024, I joined Sokoine University of Agriculture at the Department of Veterinary Medicine and Public Health. So I'm working there now. And then 2 years ago, I joined a master's degree, which I expect to finish this year. And now, just to remind you on today's topic from my side, so I will be looking on the transmission, on the role of small ruminants in transmission dynamics of African trypanosomiasis. And this will be in Kibondo district and Serengeti district. So I will talk a little bit about the study site. So maybe just the problem statement of my study is that African animal African trypanosomiasis, but also the human form of trypanosomiasis is still a problem, especially in sub-Saharan Africa. But the evidence indicates that sheep and goat can harbor the disease, and sometimes they don't show clinical signs. But now these animals have been disregarded when it comes to management and research. So you can see many publications in cattle, but not in sheep and goats. So Harriet has said the wildlife can act as the reservoirs, but also these small ruminants, sheep and goats, can also act as the reservoir of human infective trypanosomiasis or trypanosomes. So, okay, so now, why am I going to do this research? It's because of its importance in livestock, but also in humans. And as I said, the small ruminants are ignored, but now this study will investigate and generate data on trypanosomes infection, but also in small ruminants, including the human infective trypanosome, which is called the rhodesiense form of the trypanosomiasis. So from this study, I aim to develop an inclusive and evidence-based control strategies, which will also integrate different sectors and disciplines that is the One Health, and so that we can tackle this problem. Yeah. So I have 3 questions, research questions. So what is the prevalence of African animal trypanosomiasis in small ruminants? It is not very well known in Tanzania. So this is what I'm going to look. But also the second one is what is the contribution of Trypanosoma brucei and Trypanosoma brucei rhodesiense, which is the human infective trypanosome? What is the prevalence? Is it found in sheep and goats or not? But also I will look on what are the risk factors for infection in these small ruminants. So from those questions, I have these 3, the main objective, but also the specific objectives. So these 3 objectives will cover will answer my research questions. So, and just to mention on the methodology, I talked about the study area. So you can see 2 dotted two red dots. So on the top there, that is Serengeti district, and I would do in the wildlife livestock interfaces. So that is human livestock wildlife interfaces, just close to the wildlife protected areas. But in Kibondo, this is the area which also reported human African trypanosomiasis, and that's where I'm also going to look. So 2 study sites. So this will be a cross-sectional study, which will include blood sampling. So this number of sheep and goats will be sampled, and then I will also take a questionnaire wherever I'm sampling to that household. I will also be taking a questionnaire to answer my third question. So for the lab diagnosis or the lab analysis, I will do DNA extraction, and we have set just a small lab in Mugumu where we shall be doing our DNA extraction, and we're happy that you also have a PCR machine and a good lab here. So maybe in the future we can collaborate. So yeah, so and all Trypanosoma brucei positive samples we shall run the nested PCR, but also we shall use the serum resistant associated gene, which is found in human infective trips, to identify if it is present in those Trypanosoma brucei positive samples. So I will run statistical analysis, the descriptive, but also I will do multivariate and mixed defect logistic regression, just to make sure that I capture all the data and the results. So what are the expected outcomes? So I have identified 5 outcomes, which are key. And the first one will from this research, so it will be easy to identify the evidence on the role of goats and sheep in the transmission of animal African trypanosomiasis, but also human African trypanosomiasis. And also will determine the prevalence and species diversity of trypanosome species in these 2 study areas. But the third one, I will also identify the presence of human infective trypanosoma species in small ruminants. Just at the end, as I said, I will also look at the from this research, we shall identify the risk factors for infection in sheep and goats. And finally, this will inform good strategies for the control of this disease. So just from there, thank you very much. This just to highlight or to acknowledge the Sokoine University of Agriculture, University of Glasgow, and the African for the support. And also I acknowledge the Grummet Fund for welcoming us to provide this talk today. Thank you very much.
Well, good afternoon, everyone. I'm a PhD student at the University of Edinburgh. My name is Mackenzie Moore, and my PhD is focused on drug resistance. So specifically for African animal trypanosomosis, we've heard about the impacts of the human disease. I'm looking specifically at the disease that can affect livestock and wildlife. So just a quick summary of how I came to my PhD. I did a degree in medical anthropology at University of California, Berkeley, worked in international health governance, got my Master's of Science in Disease Control and Prevention, and I've worked the last couple of years as an epidemiologist and biosecurity researcher before ultimately moving to Scotland for my PhD, and now have the incredible opportunity of working in Serengeti district in Tanzania. So a quick background about drug resistance itself. You heard from Harriet that a lot of the work that they've done over the last 10 years has found that resistance is one of the primary causes of treatment failure. We only have two drugs that are widely used, diminazine acetate and isometidium chloride, for prevention and control through both chemotherapy and chemoprophylaxis. There have been increasing reports of treatment failure against both of these diseases, which is unsurprising given that they were introduced about 70 years ago and had reports of resistance or treatment failure since the 60s. But encouragingly, what would be the first drug in 70 years is currently under development. It's a benzoxaboryl group. Despite this progress, we really don't know that much about resistance. We've been fairly limited in our ability to genetically study and manipulate trypanosomes in laboratory settings, and field studies on resistance and kind of picking apart the reasons behind treatment failure are costly and time intensive, and we don't have a clear understanding about what the true prevalence of resistance is and what the spread of it looks like in the field. One of the main questions I'm looking at for my PhD is about transmission fitness. So I think we all know by now trypanosomes, both the human and the livestock version of the disease, are transmitted by the bite of the tsetse fly. For any disease that's transmitted by a vector, it's really essential for that parasite or pathogen to be able to complete its full life cycle through the arthropod vector. So for tsetse specifically, because that's what I'm working on, it means if a tsetse fly takes a blood meal from a cow, from an elephant, and that animal's infected, it ingests the parasite and it goes through a full life cycle inside of the fly. Transmission fitness means it's able to complete that full transmission, going from infected blood and being able to develop in the mouth parts or the salivary glands into the specific morphological form that can then be reinfected into another animal when it bites someone else, even if they're not infected. So transmission fitness, being the ability to complete that full life cycle, is really important for the ability of a disease to spread. My PhD is broken into two primary sections: assessing fitness cost to transmission, and then also looking at the genetic landscape of resistance in the field, which, as I said, we really don't know much about. In order to look at fitness cost, I'm working in the laboratory in Edinburgh to generate drug-resistant trypanosomes and also carry out tsetse laboratory experiments. Currently at University of Edinburgh, we're the only tsetse lab in the UK. So we have an insectary where we're able to work with Glossina morsitans. And so I, by choice, spend most of my day surrounded by tsetse flies, trying not to get bitten and injecting them with nasty things. So I was very worried coming out here that there would be a vendetta against me, of all the tsetse flies would know I'd been experimenting on their cousins, but I've not been bitten very much yet. The second portion about looking at what resistance actually is in the field, I've been working to develop a test to be able to identify drug resistance and then also actually ascertain what the prevalence is in livestock and wildlife at this livestock interface, which we've discussed is really essential for how a disease spreads and where the reservoirs exist. There's been significant work in the last couple years being able to finally identify which trypanosome genes are associated with drug resistance. So through functional genetic studies, we've been able to identify and validate 3 specific genes in the different trypanosome species. So for the livestock and wildlife disease, we have Trypanosoma brucei, Trypanosoma congolense, and Trypanosoma vivax. For TBAT1, this is a drug-resistant gene for Trypanosoma brucei, and it means that if it loses this specific gene, which means that the cell can take up the drug, it becomes resistant to diminazine acetate. CBP is relevant for all of the animal diseases and all of the animal pathogens, and is for that new class of drugs that I talked about, the benzoxaboryl, but is different from TBAT and DMT in that it activates the drug through cleavage. DMT, just like TBAT1, is a transporter, so again involved in taking up the drug into the cell to get it to work, but that's for T. congolense, for isometidium chloride. So once I'm able to generate my infectious, resistant trypanosomes in the lab by getting rid of these individual genes, I can then infect tsetse flies with them. So what our tsetse infection system looks like is feeding tsetse flies from trypanosomes that we've either grown up in culture in flasks or we've grown up in mice. Once we reach the parasitemia level that we want, we're then able to mix the parasites with defibrinated horse blood and then feed the tsetse flies on a heated membrane. So the tsetse flies we keep in individual cages. They're then fed on a silicone membrane that has blood underneath on a metal sheet, and it's heated to mimic the temperature of an animal that they would be feeding on. So they go down, they feed, they come back up. I don't know if you can see in the back, but they do get very, very full, and that's how we get them infected. So back to the big question about transmission fitness, we really want to make sure that if they're taking up trypanosomes, that trypanosome is able to migrate all the way through to its infective form in the salivary glands. So by day 14, we expect to see trypanosomes in the procyclic form in the midgut, which we can do by dissections. You can see kind of the gut lumen and these really skinny, wriggly procyclic forms. And by day 28, we expect to see the mature trypanosomes, which are the metacyclic forms in salivary exudate. How we determine that tsetse flies can now transmit infectious trypanosomes is not very glamorous. We take the tsetse fly, we put it in a single tube, and we put it on a glass slide on that same membrane they feed on. They think they're feeding, but really they just smack into a glass slide and end up spinning up all over the slide. What we can see is then tsetse spit. On the left is tsetse spit with no trypanosomes. On the right is tsetse spit with lots of trypanosomes. If you remember back, what we really want to make sure is it's these specific different morphological forms throughout the cycle. So we want to make sure what we're really capturing are metacyclic forms which can be transmitted, because there are other forms that can't be transmitted. So how we do that is by staining the slides. In the middle, you can see this is an example of a slide that's been magnified on a microscope, and we stain it. And when you stain it, it captures the different nuclear material in these different organelles that are specific to trypanosomes, the kinetoplast and the nucleus. And here on the left-hand side are the metacyclic forms, which is what we're looking for. And you can see they're pretty unique in both their morphology, the shape of the flagellum, kind of, and then really the locations of the kinetoplast and nucleus are fairly far apart. Whereas in epimastigotes, which you can also find in the salivary glands, but are not infectious, are really close together. So once we've fed our flies with infected trypanosomes, either normal trypanosomes that are susceptible to drugs or trypanosomes that we've genetically modified to be resistant, we then want to make sure that they're able to go all the way through and we can count how many of these true transmissible metacyclic forms we have. And what we have found with our drug-resistant designed lines for Trypanosoma congolense and Shrewsbury is we really don't think that there's a cost to transmission fitness, which has fairly profound implications for the epidemiological spread of trypanosomes in the field if resistant lines are able to compete as efficiently as susceptible lines when they are going through the infection cycle on feeding on different blood meals. The second portion of my PhD is about designing a test to be able to identify resistant trypanosomes in the field. I've been focusing specifically on the DMT gene, which is for Trypanosoma congolense, and have thus far optimized this test in the laboratory setting, and we're now prepared for the first time to test it in the field to use genetic analysis to try to map what trypanosome resistance looks like in the field. We'll be doing that through a field study that involves both livestock and tsetse sampling. I'm very fortunate to be working with Paul. So where Paul's looking at sheep and goats, I will be looking at cattle and what resistance prevalence looks like in them. And while we're there, also interviewing livestock holders to dig in more to the risks involved with resistance development. It will also be a cross-sectional livestock survey where our villages share borders with protected area boundaries. The tsetse sampling will be using NZ traps with transects through protected areas into farming areas. One of the findings from the work that Harriet was involved in is that we don't really see tsetse past the 5 kilometers. So keeping those boundaries in mind, And what we're really hoping to find is what is the true prevalence of resistance in the field, because we really do not know, and just get a better idea of what the risk factors are that drive the spread of resistance, whether that's the ratio of what hosts tsetse can feed on, the presence of wildlife, drug selective pressure, where if tsetse are feeding exclusively on wildlife, there's no selective drug pressure, whereas if they're in a mixed environment with livestock and wildlife, you do get that drug pressure by feeding on the cattle. So thank you so much for listening to my talk. Happy to answer any questions once we're all done. Yeah.
Thanks, Mackenzie. I'm Abigail, also a PhD student working with Harriet, and I'm working on human African trypanosomiasis in the wildlife reservoir. As Harriet was mentioning, this is a kind of important source of where potential human cases would come from. So to give some background, the human disease is caused by subspecies of Trypanosoma brucei, of which there are 3. First is TB brucei, which is actually not human infective, but does cause disease in animals. Then we have rhodesiense, which causes acute human disease, and that's found in East and Southern Africa. So that's the one in this area, which I'm interested in. And we also have gambiense, which causes a chronic form of human disease, but just found in West Africa. So brucei and rhodesiense are morphologically identical, but rhodesiense infects humans, and this is due to it having a gene called the serum resistance associated gene. And this means, yeah, it can infect both humans and animals. Brucei does not have this gene and therefore can't infect humans. And this gene is the only thing that makes rhodesiense human infective, which I think is very interesting. These 2 subspecies can recombine, so they can mate together, which can potentially cause new variants, new human infective strains. And there are as Harriet was mentioning, a history of sporadic outbreaks, potential for epidemics, and increasing human cases. We're not really sure what's going on in the wildlife. So yeah, wildlife is an important reservoir of this disease. As Harriet mentioned before, brucei has been detected in many different wildlife species, but there's some shortage of data in some of these species because, as some of you will know, wildlife are really hard to sample, and expensive and logistically difficult. And also other published studies around this in different countries have given slightly different results. So there's a need for a bit more wildlife sampling, and getting some more data to fully understand what's going on. Possible way to do this is, or hopeful way to do this, is being able to detect the parasite in wildlife feces. This has been done in cattle feces and in experimental infections by Isabel here. So if you've got any questions, you can ask her. And it's also been detected in wild chimpanzees. So the first aim of my PhD is to collect matched blood and fecal samples from wildlife, and then see how the detectability of brucei compares between the fecal and blood samples. Once that hopefully works, would then be to continue collecting feces samples from target species. So this is species that were found before to have high prevalence of brucei, and then screen for brucei using PCR, and then another PCR which we'll be able to use to detect the SRA gene in order to be able to detect any human infective parasites. Then with these samples from wildlife, we can answer some questions around what wildlife factors increase the disease risk. For example, are there certain species that will have a higher prevalence of brucei, or is species age? Does species age have an effect? Does tsetse density impact the prevalence? and also is there a high prevalence of the human infective rhodesiense in wildlife in areas closer to humans. Secondly, is looking at the genetics of T. brucei. So as I said before, mating can occur between brucei, so the human infective and non-human infective forms. But they don't have to mate to reproduce, they can reproduce clonally. Some previous work was done in humans and also cattle that showed some variation in the population structure, so whether they're mating or cloning to increase population size in rhodesiense and brucei. But no studies have been done in wildlife. So there's a gap in the knowledge here, because in humans it might often, you can see a lot of cloning, but it could be that it could be all the same variant of rhodesiense. And yeah, in order to understand a bit more about the genetics, my lab back in Glasgow have been developing using whole genome sequencing, using PacBio, which is this very cool looking machine here, which does long read sequencing at very high accuracy. So from samples collected here, I'm going to aim to sequence brucei and any rhodesiense positive samples from wildlife blood, and also see if feces samples provide enough, if the DNA is a good enough quality to be able to get some sequence data. This will enable me to answer some questions around the parasite and disease risk. For example, how much recombination, or what's the population structure of brucei and rhodesiense in wildlife? How does the human infective form spread through wildlife? Does the parasite genotype, so as I mentioned before, mating between strains could result in new variants or new strains of human infected parasites, and whether the genetics of these might mean there are some that are more virulent or spread more quickly. Also, potentially the gene that causes human infectivity can have some variability, so that have more than one variant of this gene have been discovered. So it's seeing if that will affect the severity of the disease. So I'm carrying out my sample collection in Serengeti. And the reason for this is it's a historic and ongoing focus for rhodesiense human infected form. There's other ongoing research in the area that will help me to collect samples. So to start with, I need to collect matched blood and fecal samples. And there's also a lot of historical data and research infrastructure here, and loads of wildlife, most importantly. And I'm hoping to do as much lab work as possible in the country. So I have literally brought in my suitcase some lab equipment, and the aim is to hopefully be able to set this up in Serenera. So I've got, the ability to do DNA extraction, PCR, run gels, and do qPCR if all works as to plan. So yeah, in conclusion, wildlife are a reservoir for T. brucei, and mating can occur between the human and non-human infected parasites. So to sum up my aims, first of all, to validate fecal sampling to see if we can detect brucei in wildlife. This is not part of my project, but if this works, I think fecal sampling will be a really interesting, non-invasive way of sampling, for example, for rhino or other diseases in the future. Then aiming to determine particular wildlife factors which will increase the likelihood of infection, such as species or age. And then also carry out some genetic analysis to see how the human infected parasites circulate in wildlife and how this might lead to human cases. Thank you, and thanks.
All right, thank you very much, Harriet or Mackenzie, Abigail, for very informative presentation. I know there are some people online, maybe they So for Q&A, for those who are online, you have two options. You can either type your question, then we'll read it loud. But also you can raise your virtual hand. I will select you guys, and then you can ask your question. You can just unmute yourself and ask. But I will give priority to those who are. Maybe I'll start with those who are on site, while we are still figuring out like the technology thing. All right, anyone with a question? Na mtu anataka kuuliza swali lolote kwa wataalamu wetu hapa? Yes.
Thank you for all presenters for last presentations. Very good. So, I had one question to ask. Just I don't ask. Yeah, I have one question for each. Yes, yeah. The first one was from Professor there. Right? I have some. Yes, yeah. The first, yeah, yes, yeah. Okay. You also presented very well on the role of reservoirs in Serengeti, Tanzania in general. And you showed us in the impact of surveillance, as you saw that graph. Tanzania, there is a very low detection rate. And then you left us questions. Did specify a conservationist or, I don't know, has some impact. So now, in our country, as we know, trypanosomiasis will say that one of the conservation disease that on how I've been learning now. As we saw there, buffaloes did not get the disease there. They just feed. They don't get the disease. At the same time, the disease is more in the bushwalks. I remember, yes, yeah. So now, and then introduce the method of controlling, aerial spraying. So now, if we spray the wildlife, so what did we expect, considering the role of ecology in wildlife? So if we kill the tsetse in wildlife, what's the next? So what do you recommend in that one? Do we need to just remove the tsetse in wildlife, or just protect the livestock wildlife interface? So what do you recommend that for that scenario?
That's a very good and quite big question. So if there's the question about can we do interventions in wildlife to reduce the reservoir of disease in wildlife? I think maybe no one has ever done it successfully. It's not impossible, and there are examples for other diseases. And one of the challenges with trypanosomiasis is there is no vaccine. So we cannot use vaccine approaches. Maybe one day there are some vaccine candidates which are coming. So maybe one day we could do that. Yeah. At the moment, I think you can imagine that you could trial interventions around, like, I don't know, maybe having insecticide on rubbing posts or something like that. But no one has ever tried. I think it would be very challenging. So then is there the question, do we just target our tsetse control in the protected areas and the buffer zones?
Yes.
I guess more or less that's what we do now. And tsetse control can be very effective. So really that's where a lot of the effort is going now on vector control to try and reduce disease. I think what we see now really on the edges of the protected area, obviously we know that livestock keepers will sometimes bring their livestock into the protected area, even though they are not allowed to, when the grazing is short. That is probably the time when they are most exposed to disease. But some of them will be, even if the numbers of flies are still high, they will still be exposed. So I think control around the edges of the park can help to protect the livestock keepers and the communities, but that doesn't help, for example, rangers and park staff who are within the protected areas. So I think we really need tsetse control in those protected areas as well to protect the people who are working within the wildlife and tsetse areas. So probably there is not one answer. I think we have to integrate the different control measures to protect the different populations that we need to.
Yeah, thank you.
All right. There are two people from online. Now, Richard, Yunus, you can unmute yourself and ask.
He's speaking, but can't.
So this one was working. Hapo hiyo.
Okay. Hello.
Unmute your comment. Then we get it. Yunus, can you ask your question again? We didn't hear. Yunus, can you just type your question for now while we're... You can just type your question and then maybe we can ask someone else. I don't know what's the problem. Baraka, can you help us to ask your question? Baraka? No? What's happening?
Hello, Baraka. Can you ask your question? We haven't heard Yunus. Maybe it's a technological thing. Maybe we can hear from you.
Elimu wameshia, wengine na 7, wengine wamefeli form 2, wengi form 2 na wengine wameishia 4. Mimi ni kupromise tu mzee wangu, hata mzee wangu na nafasi ya kuja kumfundishia pale Diamond pale Wasafi au Wasafi kitu sasa ili tuendelee kwenye degree. Nataka niende na mzee wangu kwenye degree kwa sababu unajua uwezo alionao Diamond Platinum. Ukimwambia kuhusu vyeti anakuwa anaona kama wewe unamchanganya, kwa sababu tayari ameshatengeneza mifumo ambayo ya kutengeneza utajiri wa hela nyingi sana.
Hi George.
Lakini sasa sisi maneno kanakuwa kanatucheka sana. Hajasoma huyo. Yaani kwa namna moja au nyingine unakuwa sasa hata kusoma elimu ndio, lakini kuna muda kana sina. Kuna kiungu.
George, maybe for now, because of the either technological challenges or something, people should just type in questions there to save a bit of time.
No, we just heard you. Did you say something?
Yes, I was advising that those who have questions, they can just type them there to save us some time.
Okay.
And get us any questions.
Yeah, fortunately now we can actually hear clearly. We just had some technical problems. Now we can hear. So I don't know who was Baraka. Did you have a question?
Yes. Good evening, everyone.
Good evening.
So my name is Mr. Baraka Edward. I'm currently working for the ECOP project in Tanzania. So my question was, now in Tanzania we have the same issue of the invasive species, so especially on the conserved areas. So what are the other impact of the invasive species rather than colonization? Maybe the issue of speeding this parasite on our conservation areas, and what can be the mitigation measures that can be adopted so as to solve the problem in Tanzania? Because we know sometimes we have our faunas that tend to move from one place to another place. So how can we mitigate this issue?
All right, Baraka, thank you very much. So your question specifically is about the invasive species, if I get it correctly.
Yes, actually it is. Yeah.
We had 4 speakers today, we had Harriet, Paul, Mackenzie, and Abigail. I'm just trying to figure out like who maybe will answer your question, but we will definitely. You have.
I'm not.
We are really not very sure if that question is for this, but we can generally just like invasive species.
Example of the species.
Do you mean plant species? Because I don't think we can answer it.
Yeah, so we are trying to figure out. Maybe you can type to be very specific so that we can answer. But meanwhile, Yunus, the person who was trying to ask the question, you can unmute yourself and ask. But meanwhile, there's a question for Paul. What are the common species of vectors reported in Kibondo DC? Maybe you have to come here because I don't know, like.
Everyone's listening.
No, they can hear you.
Okay.
If you can just answer maybe that one while we are waiting for someone to attend that. So on the chat is the question for Paul, which asking what are the common species of vectors reported in Kibondo district?
Yes, so thank you very much for the question. So in Kibondo district, especially close to the Mwewoz Game Reserve, the common species are more or less the same as reported in Serengeti district. So that's why I tried to look in the same ecological, in the areas with the same ecological features. So we have the Glossina parridipes and Glossina snotunai, which are also present in Kibondo. So, but the more prevalent, I think this is the Glossina parridipes. Yeah, thank you.
All right, thank you very much. There is another question here from Naushard Yunus. Why do you prefer to take blood sample on the animals rather than other sample? And which consideration will you take out to meet animals' welfare during the sample extraction? That's general. Like from the presentation, I'm just like trying to from the audience. So I think the last presenter, Abigail, she stated that she'll be collecting blood samples and fecal samples as well, and to see like the efficiency of the, like if they can detect disease from what. So with blood samples, why do you prefer? Is it like preference or
Compared to what?
Compared to other samples? Yeah, maybe fecal samples. So maybe the way the question was rephrased. Like,
It's like why
do you prefer to take blood samples on the rather than other samples, like maybe fecal
or whatever?
Okay, what's the parasites are mostly found in the blood?
Yeah, so the parasites are mostly found in blood, so it's much easier to detect them.
Shall I come over there?
All right.
I can add a little bit more.
Yes.
We asked also about the welfare. So we do a lot of sampling from livestock, where it is relatively straightforward to take a blood sample because the livestock keepers can help us to handle the animals. But in wildlife, that's really difficult for lots of reasons of cost and ethics and welfare and logistics. So if it's possible to use a sample like a fecal sample, then that's much easier because it's less invasive. So it's much cheaper and safer and better for the animal. So that's why we're interested to develop some of these diagnostic approaches and materials other than blood so that we can take samples from the environment more easily.
All right, thank you very much. There's a question from Meklina. Meklina, you can unmute yourself. Meklina, can you hear us?
Hello,
Hello.
can everyone hear me?
Yes, we can hear.
Yeah, so sorry, I'm not McLeaner. My name is Esther Kimaro. Talking from University of Glasgow. Hi, Harriet. Yeah, so I'm happy that McLeaner introduced me about this Zoom, and I'm here. I've been hearing your presentations. Was really nice. Just want to, first of all, thank you, Harriet, for a very nice presentation on the background of, tsetse and trypanosomiasis in Tanzania, and the great work that has been done, and where we are now, and the research gaps that we still need to, to investigate further. So thank you very much for that background. It actually helped me to refresh my understanding in the trypanosomiasis. So I don't have a question, just to praise you. The presentation was great. Also for Abigail and Mackenzie, thank you very much for a good presentation, and actually the research gaps that you have identified. I can say really those are the areas that we are still missing in the great work that has been done in Tanzania in the area of tsetse and trypanosomiasis. Actually, I've done some work in this area. This is another area of my research interest. So I was happy to hear the further work that needs to be done in this area. So for Paul, are you there?
Yes.
Yeah, he's here.
Yeah, happy to hear your presentation. I know we have been talking about this before, but I just have few points just to improve your work, especially in the presentation. I was looking at your title. You talk about the role of transmission for the small ruminants, but I would advise you to try to align it very well with the gap, because, sorry, with the main goal. In the main goal, you talk about the impact. So I think the reflection from the title has to align with the main goal. Actually, the impact, it can be a bit controversial word. It's something different. I think focusing on the role of transmission for the small ruminants will be much better than talking about the impact. In your main goal, we talk about the impact of these small ruminants in the transmission of sleeping sickness. So that one, you try to improve that, the cohesion between the two. Another thing is about you are planning to do your study in Serengeti and in Kibondo, but in your explanation, you didn't really show clearly why do you want to do this study in two different places. That one you didn't really explain properly, like what is the aim of doing that? What do you want to achieve? Why having these two different areas? I heard you talking about the tsetse species. Someone asked something like that, but it was not very clear in your presentation. So the same in your document, that one has to be clear somewhere, starting from your background information, from the problem statement. That one has to feature properly so that someone can see the justification for you to do this study in two different areas. Apart from that, the One Health, the expected outcome for the One Health-based surveillance, I would love to know how are you going to achieve that. If you can, maybe for that one, I'd love to hear a response for that, the One Health-based surveillance as your expected outcome for this study. If you can comment a little bit, because I didn't get how are you going to achieve that out of this study. Thank you very much.
Awesome. So the last question still to Paul.
Well, thank you very much, Esther, for your inputs. And yeah, so just to answer the question on the One Health-based control strategies. So from this research, so the findings will inform for better One Health-based control strategies. So just from the research output or the research, so the research findings, so I will write like a policy brief just to recommend for One Health surveillance or control strategies, which will include people working in the wildlife conservation areas, the animal health professions, but also the human health workers, without forgetting the environmental health workers. Thank you very much, and I appreciate your inputs, but I think some of the things you said, because of the short time I had, I didn't explain. Yeah, thank you very much.
Thank you very much. And Harriet, I'm looking forward to meet you when you come back to Glasgow. Thank you very much.
Thanks. All right. So for those who are here, anyone has like a comment or a question to our presenters? I've definitely learned a lot. And any comments from that?
I think that's people sampling for other.
Yeah, kind of. We can help with that. Yeah. Okay. Take a note.
It'll be very interesting to see.
Yeah. Oh.
Okay.
Yeah.
Just a comment to presenters. Thank you so much for the nice presentation. Then on behalf of the grooming teams and the other staffs around here, we appreciate for the knowledge you bring us. I think when the coming days, I'm the one because I have the wish to start on such on those mostly animal zoonosis, zoonotic diseases which may be transmitted from wildlife to livestock. But that's a topic of my test. I think I'm so happy to hear from you and for the aspiration. I think the coming days, just a few days to come, I think it's something big.
Thank you very much.
Thank you so much, guys.
Yeah.
We're very happy to discuss afterwards with anyone who has more questions.
There was one last question from Mabenga Peter. This question is to any presenter. Is vector control strategy one of the intervention to consider in Tanzanian context?
Vector control.
Vector control strategy.
Yes. I think vector control always has to be part of our strategies for trypanosomiasis control, whether that's vector control within affected areas, for example, with targets, or whether it's vector control through what farmers are doing with treating their livestock for insecticide. There's some other work that I didn't present today because I thought I'd talked about enough different models, but there is another model we have done which looks at the best control strategies to reduce the risk of resistance when livestock keepers are using trypanosides. And actually we find these integrated control strategies are really important. So using some insecticide control also reduces the risk of resistance emerging to trypanosides. So yes, I think vector control is absolutely key, but there can be lots of different forms of vector control and we have to choose the right type for the right place.
All right. So thank you very much. I'll say bye to our online participants. And then I really appreciate, and we'll keep you informed about our next seminars. Thank you.